Hello, my name is Jesse Rodriguez, and I'll be doing my presentation on CRISPR-Cas9. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And for the past few years, the CRISPR-Cas9 system has become a popular method of genome editing because it's fast, cheap, precise, and relatively easy to use. So genome editing is a method that lets scientists change the DNA of many organisms, including plants, bacteria, and animals. This is done by targeting specific DNA and splicing it in order to make insertions or deletions right in the organism's genome. Editing DNA can lead to changes in physical traits like eye color. The way it works is that researchers create a piece of RNA with a guide sequence, which is complementary to a targeted bit of DNA in the host's genome. So in other words, if the DNA has a sequence that reads 5' GGCTAT 3', then the RNA guide sequence is exactly the opposite and reads 3' CCGAUA 5'. Also, if you notice, there is a U for uracil instead of a T for thymidine because the guide sequence is made of RNA and not DNA. The Cas9 protein then cuts the DNA and attaches to the RNA, and the whole thing binds to the targeted DNA sequence in the host genome. The Cas9 RNA complex then makes a double strand cut in the genomic DNA, and an alternative piece of DNA can be spliced in right at that spot. CRISPR can also be used to study and prevent diseases as well as generate new tissues from stem cells, which are used to regenerate or repair diseased tissue and organs like heart muscle tissue and neuronal tissue. It's also possible to treat a whole multicellular organism with genome editing. For example, a mouse with liver disease had a genetic defect that was treated with a CRISPR-Cas9 mediated genetic change, and it improved the mouse's symptoms. One important point to note about this mouse example is that the change was made to somatic cells rather than germline cells, meaning these genomic modifications aren't passed to the next generation. That said, CRISPR-Cas9 technology is able to alter the DNA in germline cells, and if that's done, then the engineered changes can be transmitted across generations. This has been done in several organisms, including mice, monkeys, and most recently in humans. So a couple years ago, a Chinese scientist named He Jiankui claimed at a conference that he had edited the genes of twin girls using CRISPR-Cas9 technology. He specifically altered the CCR5 gene, making the carrier resistant to some strains of HIV. He made the changes in 2 embryos which were then implanted in a woman and carried to full term. The announcement led to an international moral outcry. First off, Jiankui acted in secrecy and did not tell his institution about his experiments. Second, the procedure didn't have a known medical need since neither of the infants had an HIV infection, and the procedure inactivated a healthy gene. Third, genome editing technology is still in its infancy, and there's no global consensus about using gene editing technology on human subjects. The risks of doing this to human embryos remains difficult to predict. For example, according to Jiankui's presentation, not every cell in embryos was edited by CRISPR-Cas9. Also, the mutation made for the CCR5 gene didn't exactly match the known variation of the gene that confers HIV resistance. This means it's not clear if these untested changes will provide the same benefits or introduce new unintended consequences. The general consensus is that we're not there yet, and that the scientists have moved quicker than the ethicists. So as you can see, CRISPR-Cas9 technology has feasibility in human germline genome editing, but still raises very complex social, ethical, and legal questions about questions that we need to answer before we go any further. So here's a comic I found. It's kind of funny. Looks like an affluent couple looking window shopping for babies and deciding if they need a tech tycoon baby or a tax lawyer baby.